I would like to share my research with you today. The title is A Novel Approach to Treat Osteoporosis, Bone-Targeted Delivery of Heparan Sulfate. Well, you may not notice it, but our bone is a quite dynamic tissue that is constantly undergoing old bone broken down and new bone formation. Excessive bone broken down leads to osteoporosis. More than 200 million people are suffering from osteoporosis worldwide. Furthermore, 1 in 3 women and in 5 men over the age of 50 years will experience osteoporosis-related fractures in their lifetime. So how is bone broken down? This picture shows an osteoclast, which is the only cell in our body that breaks down bone. It sits on bone surface and secretes various enzymes. And among them, Cathepsin K is the most important one because it degrades collagen, which makes up about 90% of the bone matrix. Since Cathepsin K is the major player in bone degradation, many scientists have been exploring the methods to inhibit Cathepsin K activity for a long time. To date, several Cathepsin K inhibitors have been developed, but they all failed to enter the market because of various side effects. Reasons for the side effects were pointed to 2 aspects. One was inhibition of Cathepsin K activity towards non-collagen proteins. The other one was undesirable Cathepsin K inhibition in non-bone sites. Therefore, it is of great importance to develop a novel Cathepsin K inhibitor. A recent study showed that a linear polysaccharide called heparan sulfate can selectively inhibit the activity of Cathepsin K towards collagen. Heparan sulfate is composed of a repeating disaccharide unit. The R groups can be replaced by many functional groups. So in nature, heparan sulfate can have different structures and sizes. I hypothesized that bone-targeted delivery of a heparan sulfate-based molecule has the potential to treat osteoporosis with fewer side effects. Since heparan sulfate can have different structures and sizes, I first screened 52 kinds of heparan sulfate and found the heparan sulfate that can inhibit Cathepsin K the best. With that in mind, I then wanted to deliver this heparan sulfate to bone tissue specifically. This will be achieved by attaching a small peptide with 7 amino acid D to heparan sulfate. Repeating sequence of amino acid D has been shown to bind to hydroxyapatite with high affinity, and hydroxyapatite only exists in bone and teeth. To test the efficacy of this peptide, I injected this peptide that was attached to a green dye or the green dye alone into mice and collected their bones for color detection. We can see that mouse bones showed strong green color 1 day after peptide-attached green dye injections, indicating these peptides successfully bound to bone. I didn't see any color from mice injected with green dye alone. To find out how long this peptide can stay in mice bodies, I monitored the intensity of the green dye for a week. The green dye was gradually lost in bone. Nevertheless, comparing to day 1, the color intensity still remained almost 60% on day 7. This is ideal for a drug because when you take a drug, you want the drug to stay in the body long enough to function before your body clears it out. My next step is to use this peptide attached to heparan sulfate to treat mice with osteoporosis. To sum up, based on the data from this project, D7 heparan sulfate can potentially be used as a novel osteoporosis therapeutic. It may overcome the shortcomings of side effect-prone traditional Cathepsin K inhibitors, which all failed in clinical trials. This novel approach could benefit millions of osteoporosis patients worldwide. So far, our animal study of this novel approach has showed very promising results. Since heparan sulfate is expensive to synthesize, my future plan is to develop an antibody that mimics the function of heparan sulfate to treat osteoporosis, because antibodies are cheaper and easier to produce on a larger scale. If you have any questions, please feel free to contact me. Thank you very much.